Hello, everybody. So I'm Ivo Boneca, one of the co-organizers of this MOOC, and this is my colleague.
I'm Grégoire Chevalier, so we are both the co-directors of this MOOC, and today we're going to answer your questions. The one that we got from the discussion, or the one that you can ask live.
Okay. So are there any burning questions, or should we start trying to answer from the forum the questions?
So first, maybe just to let you some time to ask questions, we noticed that you had a very intense debate over ACTH and whether or not it's a neurotransmitter or an hormone. So I know that a lot of you were a bit disappointed because maybe the question wasn't really well asked, but actually the answer is both, because of course ACTH, actually ACTH stands for hormone, but since it's produced by neurons and it's going to signal to other cells, it's also a neuromediator. So both answers were true, and that was a mistake probably of the question that was not really well asked. But just to let you know that ACTH is both a hormone and a neuromediator.
But not a neurotransmitter.
Not a neurotransmitter, because it doesn't transmit from one neuron to another.
Okay. We're waiting to see if anybody wants... you're doing great, thank you. If there's no questions so far...
Hi, Danielle.
Hello.
Hello. Do you have any questions?
Alors, je m'excuse, mais j'ai un problème. Je pense que je vais devoir quitter. Je ne parle pas anglais. Je fais tout traduire par Google Traduction, en fait. Tous mes cours.
Ah, d'accord.
Mais vous pouvez quand même poser votre question si vous avez des questions.
Non, parce que j'ai lu les questions posées par ailleurs. Et pour moi, en fait, j'ai suivi cette formation parce que je suis conseillère en nutrition et que l'étude du microbiote m'intéressait beaucoup. Voilà.
D'accord. Très bien.
Donc, est-ce que tout l'entretien se fait alors en anglais, je suppose?
Oui. Comme c'est un MOOC international.
Ouais, bah oui. Bon, bah alors, bonne conférence à vous. Et
Merci.
Merci.
Au revoir.
Au revoir. So we just had a bit of a discussion, since we know that a lot of you are French, but we are speaking in English because it's an international MOOC. So for all people all over the world to be able to understand this MOOC, we're going to talk in English and answer in English as much as we can. So we got a question about rabies virus. Okay. Indeed, we had a big question on the forum about rabies virus and blocking antibody and how it works. So we are not virologists.
No.
Hervé Bourri, with the one who did this session on rabies virus, is a virologist of the Institut Pasteur. He's specialized on the rabies virus. And what I can tell you is that usually when we give a shot of vaccine after someone has been bitten and has been infected with the virus, the idea is to win a race against the virus. The virus is going to spread from the part where you've been bitten, and it's going to go up to the brain. And in the meantime, so this can take up to 2 weeks or a month. In the meantime, the immune response, thanks to the vaccine, is going to be mounted and could stop the virus spreading to the brain. So in the case of this, you're asking a question about a patient that has been treated in United States, freezing the brain, waiting for the immune system to do its work. So they were not really freezing the brain, if I remember well. I think they were cooling down the body. So it was hypothermia. The idea was to slow down the propagation of the virus up to the brain. And the idea was also to win this race against the virus. So giving some time for the immune response to fight the virus?
Yeah, because in general it takes minimum 8 to 10 days to start having an efficient antibody response against the vaccine, and so that's a win. So if we are over, if the virus is spread fast enough and we don't have enough immune response that is mounted, the immune system is not able to cope with the virus. We won't have enough antibodies or robust response, immune response against the virus before it does its damage. So that's why there's a window of opportunity between the spreading and replication of the virus and the mounting the immune response. But that doesn't work with all viruses or all infectious diseases. It's a particular case, rabies.
Just to let you know, I think that in this precise case, it was probably the only patient that survived rabies infection. I think so. I have to check, but this patient survived, but in such a state. I mean, the brain was really devastated by the virus. So even if this patient survived, she was severely handicapped. So don't hesitate to ask questions. We kept some others about fecal microbiota transplantation.
Yeah. So in this case, the question was how to standardize or normalize fecal transplantation from humans to humans or from humans to animal models like the mouse. And we do agree that it's extremely complicated. There's no universal way of normalizing that. And the best way of going about it is to have a repertoire as best as possible of the microbial diversity and the input, using usually the easiest way is using 16S RNA, and then checking after a certain time whether that has been maintained in the adopted host. But there's no universal way of normalizing it, because it's extremely difficult to have to normalize in terms of amount of bacteria. There's no universal probe that would allow us to do that. So that's something that is being a problem in the field, obviously.
So technically, just to let you know how it's done, today, nowadays, the only case where we do fecal microbiota transplantation in humans is just for Clostridium difficile infection. So that's a very particular case. In this infection, it's very hard to get rid of this bacteria, even through several rounds of antibiotics. And people suffer usually chronically from this infection. And it has been shown with more than 90% of efficiency, yes, that fecal microbiota transplantation could work. And so you give antibiotics to the patients to get rid of most of the microbiota, and then through I think it's from sold. I don't know how to translate that. A probe, yes. Then you can inseminate it with a new microbiota.
And that usually helps. The normal microbiota helps to exclude Clostridium difficile from the niche, because they're very efficient in outcompeting in those conditions Clostridium difficile, which antibiotics do not do as well.
Exactly.
Okay.
Another question from the forum.
Yeah.
So we had a question on the forum regarding the microbiota of Drosophila and compared to the microbiota of the squid.
Because it's an example that was given in that presentation. And so so the person actually is raising the issue whether it's comparable or not to other animals, the squid situation. And I mean, the squid and the unique squid interaction between Vibrio fischeri and the squid is unique to the light organ. That doesn't mean that there are not other niches in the squid where you will have a diverse microbiota like you would encounter in Drosophila, or in not as diverse as in mammals, but at least as other invertebrates. So it would be more comparable to what would be a microniche like the crypts in our colon that might be microniches where you will have one or two species that are highly adapted to that. Or, for example, in the case of the human stomach, where you will have the human stomach is extremely adapted to the colonization by Helicobacter pylori. So it would be more comparable to that situation. That doesn't mean that there are not other bacteria living on other niches of the squid.
Right. To take another question from the forum, we had a question regarding the decrease of bacterial diversity with age, especially after 65, 70, and how these changes are due to lifestyle or to people getting old, and what could be the cause of this reduced diversity. So just to let you know, there have been a study that had been published 1 or 2 weeks ago in China that showed that old people, we're talking about after 90 years old, that were in very good health age, had a microbiota that was pretty similar to the one of middle age, 30, 40 people, 40 years old people. The problem with this kind of study that, of course, it shows that microbiota seems to be a good biomarker for healthy aging, as we say. But of course, when you age healthy, you don't have the same habits than if you are retired in some hospitals, if you're hospitalized very often. So it's still difficult to know what are all the causes of this good diversity in these healthy people, healthy aged people.
It's difficult to know which is the hen and which is the egg.
Exactly.
In these situations. So there's a question about the yogurt complement with live microorganisms to shuffle around the gut microbiota. So first of all, a definition of yogurt. Yogurt, by definition, is milk that has been fermented with 2 species of bacteria, Streptococcus thermophilus and Lactobacillus bulgaricus. And so those 2 species, so to call it yogurt, you can only use those 2 species. And those 2 species are only transient inhabitants of the gut microbiota. So while you're taking a yogurt, you might have shifts, for example, in the populations of lactobacilli because you're putting a new one there, but it will not stay there. If you stop eating your yogurt, it will disappear and you'll get back to the normal gut microbiota. And that's usually the problem with yogurts or other fermented milk products, is that those species are very good in fermenting those in milk products, but they are not good colonizers of the gut. And so they will transiently shift, and mainly in what usually happens is that they shift the proportions of lactobacilli, but not the rest of the microbiota. They usually compete with other lactobacilli because they're competing for the same niche, but usually if you don't give a continuous input, they will be excluded once you stop eating your yogurt by the resident lactobacilli, which are much more fit to live in our gut.
Great. Other questions? Otherwise, I can check on the forum the ones that we did not answer yet. So we can put Lactobacillus plantarum. Oh, so yes, we can.
Yeah, of course.
Plantarum is a very good so-called probiotic, especially because it has been shown also to have some specific effect on growth, general growth. I don't know if you follow the work of François Lelier, for example, who is working in Lyon at l'École Normale Supérieure. So he has shown that regarding this very specific strain of probiotic in mice and Drosophila actually, but in mice that have been undernourished, so they've got a special diet which is a bit hypocaloric. If you give this probiotic, you will have more sustained growth than mice that have a normal microbiota.
Mm-hmm. But again, I mean, it requires continuous input of the bacteria. Otherwise, L. plantarum will not stay in the gut permanently.
All the probiotics.
In general, probiotics have a hard time, which is, to be provocative, is beneficial for those who produce them because you need to take them all the time. So it's economically more sound as a
interesting.
Yeah, interesting for those who sell them. So in general, probiotics have beneficial effects, but do not stay permanently and are usually outcompeted by the microbiota.
Exactly. I'm looking for the questions on the discussion, but please feel free to ask. I think we answered most of the questions
that we had.
Not all. Checking again.
Yeah.
So maybe it was you, but someone asked a lot of questions on the rabies virus. So I'm not going to go through all of them, but I answered them on the forum regarding especially neutralizing antibodies. All the answers are already there, and I think that's it.
About the afferent nerve, no.
About the afferent nerve, that's, yes, a question for Jean-Marc Cavaillon.
Yeah.
Want to...
Yeah, somebody was questioning about the release of inflammatory cytokines from the afferent vagus nerve, and so that is not the case. I mean, the afferent vagus nerve does not release cytokines, but it reacts to them that produced in its environment, and so it can transmit that information and indirect information to the brain that will eventually feed back with response that induces an anti-inflammatory response. So that's how the afferent nerve, the vagus nerve, will react to an inflammatory response, but it's not the nerve that is producing inflammatory cytokines.
That's something that you're going to see this week, actually. Since you're going to have two talks from Ilana Gamani and Maud Pascal specifically on this topic of the vagus nerve and the enteric nervous system and how it communicates with the immune system and the brain.
And with the microflora.
And of course. So we've got another question here, just over there.
Yep.
What about gut microbiota as therapeutic in the near future? So that's a question we get a lot in conferences and congresses, because as you probably all know, there's a big fuzz around microbiota and the therapeutic potential of microbiota. So the problem we got nowadays, that in my feeling at least, is that it's pretty, we are at the early age of microbiota as a therapeutic tool, since we don't know exactly what we are transplanting. What I mean is that what you've got in your microbiota may be very beneficial for you, but it might not be the same for someone else.
Mm-hmm.
There's no such thing as good bacteria and bad bacteria. They are good if they are with you and they don't harm you, but they can harm someone else. So if we want to manipulate the microbiota for therapy, we should find kind of a standard of bacteria and fungi and viruses that are beneficial for all of us, that without any restriction.
Mm-hmm.
Because we know that sometimes if you transplant one microbiota from one person to another one, it can be kind of dangerous because some strains might be harmful for some others.
Mm-hmm. And then a follow-up question is about that gut microbiota within the family. So skinny siblings next to an obese sibling in the same household family. So I mean, it's dangerous nevertheless, even in the same family. A very good example of a bacteria that is part of what we could consider somehow our microbiota. It's in a follow-up of what Grégoire just said. Is, for example, Helicobacter pylori. So for most people, Helicobacter pylori will live during their whole life in their stomach without any consequences. And depending in the consequences of having Helicobacter will be related to genetic susceptibility. So one person will happily live with Helicobacter for their lives, and another one will have a genetic polymorphism that will lead predisposition and will lead either to gastric ulcer or duodenal ulcer or actually gastric cancer. And so it's very dangerous it's difficult to predict what will be the outcome of having therapeutic transplantation of microbiota from one person to the other. I think what we will benefit in the future is from those cases where we know how it works and why it's beneficial, is try to reproduce that by other means than transplanting the microbiota. It's trying to mimic what the microbiota is doing by other means, like with prebiotics or things like that, where we can control what we're giving and what will be the input in, and combine that with genetics susceptibility or genetic haplotypes, and knowing what are those that are associated with risk with certain diseases, like, for example, chronic inflammatory diseases of the bowel, which in certain conditions, so for a certain person giving that microbiota wouldn't make any difference, and for another one who has a high risk of genetic risk of developing, giving that microbiota will develop a Crohn's disease or ulcerative colitis.
So, as you suggested, the other mean is the diet. Ivo just talked about prebiotics. Indeed, prebiotics is a way through the diet to impact the microbiota and some strains. So, prebiotics, the definition of prebiotics is just some compounds that are going to help probiotics to grow or take a better place in the ecological ecosystem of the gut. So, and just to come back about the question with siblings and family, nevertheless, in the case that I took before, which is Clostridium difficile infection, when you got fecal microbiota transplantation, what we do usually is that we take the microbiota from someone that is the closer, so either the husband, the wife, or someone from the same house, because microbiota from people that live together is much more similar than the microbiota from people that are complete stranger. Just to let you know. So yes, diet is a way to potentially impact prebiotics. You had a question with microbiota plus gene background or yeah, indeed. So this is exactly what Ivo said regarding the genetic background that may predispose some people with some microbiota to develop inflammatory diseases, autoimmune diseases, allergy, maybe even psychiatric diseases. We don't know yet, but that's true that for a long time we thought that environment plus gene makes the disease or makes the predisposition to the disease. You are genetically predisposed to get the disease, you get an environmental trigger that gets you sick. Now there's the third level, which is kind of environmental, but which is also a bit different, which is a microbiota.
Okay.
What was the question? I think
we covered most of the questions that were in the forum.
I think so.
And
another one. So how about the mother vaginal microbiota make a big difference versus C-section?
So that's a very controversial. I mean, there are people who have been studying and they're still going on studies, but what the studies seem to show is that indeed a baby that is delivered by C-section will have initially a microbiota that is much similar to the microbiota of our skin, and then a baby that is delivered by vaginal delivery will have a microbiota that is similar to the vaginal microbiota and the intestinal microbiota of the mother. But that is very transient, apparently, and very fast. The baby that was delivered by C-section will acquire a microbiota that is very similar to the intestinal microbiota of his mother, and so on. But nevertheless, it's a question of timing, and the simple fact that the baby by C-section might have for a certain period, but might be critical in establishing homeostasis of the immune system, maturation of the immune system. And if that especially during weaning, which is seen a lot, at least well studied in mice, but it seems to be the same in humans. And if that might impact how the immune system is trained by the microbiota. And if during that period, instead of having an intestinal-like microbiota, it's still similar to a skin microbiota because it was delivered by C-section, there might be differences in the outcome in terms of how that person will react later on in terms of educating its immune system. But it's still an ongoing.
It's very controversial because there's a lot of studies that say that there is a difference in the microbiota of those babies after 6 months and 1 year, the one that have been delivered by C-section. But there are also 1 or 2 studies that did not find any difference. So, I mean, they are more in favor of a difference, but we don't know for sure yet.
Well, okay.
How the gut microbiota is ordering the item on the menu, herbivore, omnivore,
etc.
etc. Cat can sense the level of protein in the food and fat content? Question mark.
That's a very good question.
But you answer that.
So it's highly controversial. I don't think we know enough about it to know if whether our microbiota would be. I mean,
if it's regarding, I don't know if I understand well the question. If it's how the microbiota can favor our diet and what we want to eat,
I think it's
that. So because if it's this question, Sergei Fetisov is going to make a special session on that, and how the gut microbiota can impact the satiety circuits in our brain and can make us hungry or starving some kind of food.
But that's more the type of diet than satiety.
Yeah.
So,
that's something that is really interesting.
It's interesting. I don't think that there are many people working on that. There's some examples where there's adaptation. We know that there's adaptation of the microbiota to the certain type of diet. For example, Japanese have these, because they use a lot in their cuisine, algae.
Algae?
Yeah, algae. And so they have this species, bacterial species, that has adapted to digest those algae. And it's rarely found elsewhere. So that tells you that at least those people will be adapted in digesting certain foods. So they will be less in discomfort, for example, if they're eating it, while somebody who's not adapted might have a lot of discomfort because it's not able to process that food properly. That happens. So one of the things that is clear is that you might have stomach ache or discomfort or diarrhea because you're eating some diet that is not adapted to your microbiota. But so that will eventually, obviously, tilt and push you to eat something that you feel that you can digest properly. So that's one way, indirect way of favoring some diet over another one. But there might be more subtle ways than just what we know.
Yes.
It's still to be discovered. It's a very good question. Anyway.
So one last question? Or?
Well, you're welcome.
If we answer all the questions,
I think we're gonna wrap up today for a hangout, and
we're gonna do another one probably 2 weeks from now.
And
regarding
the next 2 weeks, so week 3 and 4 of the MOOC.
So thank you for
all the questions. Interesting questions. And
so see you for the next one.
Time. Okay.
Bye.
Bye.